Hello friends, welcome back to my YouTube channel Nasrin Padasale. So this is a part 12 video. So currently we are revising for our GATE examination. So first we'll be studying about the comparative difference between bacteria, archaea and eukarya. First we'll be studying about organization of the genetic material. So in bacteria and archaea there is no true membrane bounded nucleus, but in eukarya have a true membrane bounded nucleus. Nuclear membrane is present only in eukaryotes. Okay. Next in bacteria and archaea the DNA is not complex with histone, but in eukaryotes DNA is complex with the histone protein. Okay, so chromosomes in bacteria are usually one, and at the same time the chromosome will be circular in shape. This is same for archaea, but in eukaryote there will be more than one chromosome, and the chromosome will be linear. Okay, next plasmids are very common in bacteria and archaea, but in eukaryotes is very rare. But in bacteria and archaea there is no introns present in gene, but in eukaryotes you can able to find a huge amount of introns. Okay, and next with regarding to this nucleolus is not present in bacteria and archaea, but it is present in eukaryotes. Next with regarding to cellular organelles, mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus are absent in bacteria and archaea, but it is present in eukaryotes. Regarding to plasma membrane lipids, bacteria will be having a ester-linked phospholipid. At the same time, hoponoids are present. Hoponoids are nothing but they are membrane components that are involved in regulating the membrane fluidity and stability. So this hoponoids will help the bacteria to withstand any kind of stress. So these hoponoids are present in bacteria, at the same time it is present in plants. Okay, and lichens. Okay, but in archaea the plasma membrane lipids include the glycerol diether, diglycerol tetraether, and in addition to that some will be having sterols in that. In eukaryotes the plasma membrane lipids will be ester linked phospholipids similar to that of bacteria. So bacteria will be having ester link and archaea will be having ether linked phospholipid. Regarding to the flagella, so mostly in bacteria and archaea, the flagella will be submicroscopic in size and it will be composed of only one protein fiber. But in eukaryotes, it will be microscopic in size, and then each flagella will be made up of 20 microtubule in this particular 9+2 pattern. Okay. Next, with regarding the peptidoglycan is present only in the cell wall of bacteria. It is absent in archaea. At the same time, it is absent in eukaryotes. Next is the ribosome size. In archaea and bacteria, ribosomal size is 70S, and eukaryote it is 80S. So lysosome is not present in bacteria and archaea, but it is present in this particular eukaryotes. Okay, and the same cytoskeleton. So cytoskeleton is a mechanical support. It provides mechanical support. It maintains the shape. At the same time, cytoskeleton is very important at the cell division movement. So initially they didn't find any cytoskeleton in bacteria, but recent research saying that cytoskeleton also present in bacteria. Okay, so rudimentary cytoskeleton is present in bacteria and eukaryotes. So regarding to this vesicular gas vacuole, which will be providing a buoyancy to cell by decreasing the overall cell density, is present only in bacteria and eukaryotes, but it is absent in it is only present only in bacteria and archaea, but in eukaryotes there is no gas vesicles being present. Okay. So now we'll be studying the prokaryotic cell structures. Okay, so first is plasma membrane, which is a selectively permeable membrane. At the same time, it will be acting as a mechanical boundary, and it is responsible for nutrient transport and waste transport. Okay, and in plasma membrane, there will be many metabolic processes will be taking place, like your respiration and photosynthesis. At the same time, this plasma membrane also responsible for detecting the environmental clues for chemotaxis. Okay, next is gas vacuoles, which is providing a buoyancy for floating in aquatic environment. Okay, so next is ribosomes, which is responsible for protein synthesis, and next is inclusion bodies. In this inclusion body, the main role is that storage of carbon, phosphate, and other substances. Next is nucleoid, where the location of genetic material will be in this particular nucleoid only. Periplasmic space, this particular periplasmic space will be having a hydrolytic enzyme, and also it have various proteins that are responsible for nutrient processing and uptake of nutrients. So this periplasmic space is very important structure. The cell wall, it is responsible for providing shape. At the same time, it will be protecting the particular cell from various kind of osmotic stress. Capsules and slime layer, these are responsible for resisting the phagocytosis. At the same time, these capsule or slime layer are responsible for adherence of bacteria to any kind of surfaces. Next is membrane and pile, which is responsible for the attachment of surfaces. At the same time, pile are responsible for bacterial mating. Flagella are responsible for cellular movement, and the endospores are responsible for survival under harsh condition. Okay, so you can expect match the following from this particular thing. So now we'll be studying a eukaryotic organelles. Okay. So first is plasma membrane. So it will be acting as a mechanical cell boundary, and it is selectively permeable barrier with all kind of transmose system. This plasma membrane also responsible for mediating cell to cell interaction, secretions, and the adhesion of the eukaryotic organelle to particular surfaces. Okay. Next is cytoplasmic matrix, which is an environmental for various other organelles and the location of many other metabolic process. Okay. Next is microfilament, intermediate filament, and microtubule. So these are responsible for the cell structure and the maintaining the cell structure and the movement of cell. At the same time, they form a cytoskeleton, which is responsible for cell division and the cellular movement. Next, endoplasmic reticulum, it is responsible for protein lipid synthesis. At the same time, it is also acting as a transport medium for transporting various kind of materials. So ribosomes, where protein synthesis will be taking place, and Golgi apparatus, where the packaging and secretion of various thing will be done in this particular Golgi apparatus only. At the same time, lysosome formation will also be in this particular Golgi apparatus only. Lysosomes are responsible for intracellular digestion. Okay, and mitochondria, this is used for energy production to the use of the citric acid cycle, electron transport, oxidative phosphorylation. Next is chloroplast. Ultimately, chloroplast, which have a pigment called chlorophyll, which will be trapping your solar energy, so which will be helping the synthesis of food in all green plant. Okay, so they are responsible for photosynthesis, and they are also responsible for forming this carbohydrate, which is the principal food component from the carbon dioxide and water. Nucleus, it is a control center of cell, and it is also a repository for genetic information. And nucleolus, in this nucleolus only your rRNA synthesis will be done, and the ribosome constructions will also be done in this particular nucleolus. Next is cell wall and pedicel, which will be strengthen the cell. At the same time, it will be giving a shape to a cell. Okay. Next is cilia and flagella, which is responsible for cellular movement, and vacuole, which is acting as a temporary storage and transport, and food vacuole helpful for digestion, and contractile vacuole is helpful for water balance. Okay. So this is what this for this particular video. So we will be continuing. So all the best for your exam, friends. So please do watch the other part of the video. I have published totally 11 parts of this particular revision series. Thank you, friends. Thanks for watching this video.